This is the second part of the Advanced Cell Biology Stem Cells lecture. And what I'm now going to go through, after previously covering the Wnt signalling pathway, we're now going to cover the Notch pathway. So Notch is a very unusual signalling pathway, very primitive in that there's no second messenger system, no complicated signalling pathway as we've seen for the Wnt pathway. So what we have is a Notch receptor, which is a transmembrane protein, and so Notch is the receptor, and then there are some ligands which are called either delta or jagged, and you can look into where those names come from on Omin if you wish. Now this signalling pathway controls cellular differentiation, and it is a cell-to-cell direct contact signalling pathway. So whereas the Wnt pathway, the secreted glycoproteins could travel a small distance, here you need direct cell-to-cell contact because delta and jagged are on the surface of an adjacent cell. The way the system works is Notch is the receptor, delta jagged is the ligand, and when delta or jagged binds to Notch, we then get a cleavage, and the important cleavage is this one here, the intracellular cleavage, and this cleaves what we call the Notch intracellular domain to then can float away and make its way to the nucleus, and it acts as a transcription factor activator to drive gene expression. Now this is in more detail. So as the delta and jagged binds to Notch on a neighbouring cell, we get the intracellular cleavage site here, releasing the Notch intracellular domain, and this Notch intracellular domain moves to the nucleus and activates our transcription. And this only happens in neighbouring cells. So this delta signal comes from a neighbouring cell, and this often stops the same cell responding to the same signal, which we call lateral inhibition. And lateral inhibition is a central process involving tissue patterning. So it allows one cell to become one thing and another cell to become something else, but both cells would have received the same signal. So this is sort of illustrated here, where here we have in very early embryonic development some very unspecified, undifferentiated cells. This cell in the centre receives a signal telling it to be something, and when it receives that signal it expresses delta, and in doing so it signals to all of the surrounding cells to tell them not to be the same thing. So this cell has become a developing nerve cell. So this is sort of looking at how cells differentiate into different cell lineages very early on in development. So let's say this cell has become a nerve cell, and all of the cells surrounding that become something else because this cell has passed on a delta signal. These cells surrounding it have a Notch receptor, so this cell becomes the developing nerve cell, the rest of the cells become something else. So we call this lateral inhibition, that this cell says, I'm doing this job, you are doing something else. Now one of the best examples of lateral inhibition is in vascular patterning, and this is utterly dependent upon Notch signalling. So this is a vascular endothelial cell, it's a cell that lines blood vessels. These cells respond to the vascular endothelial growth factor, VEGF. And VEGF is produced in response to a hypoxic region. So when cells are hypoxic, low levels of oxygen, they secrete VEGF, requesting that blood vessels grow towards the hypoxic area and feed that region of tissue with blood and oxygen. So some cells have secreted some VEGF because they're hypoxic. That lands on an endothelial cell, and it binds to the vascular endothelial growth factor receptor. That signaling drives this cell to become a DLL4, Deltalike ligand 4 expressing cell. Deltalike ligand 4 is expressed on the cell surface. That will bind to Notch 1 on all adjacent cells. And these cells now, in binding DLL4 to Notch, we get this enzyme called gamma secretase that cleaves the notch intracellular domain, there's another one here called TACE, don't worry about that one. So the notch intracellular domain goes to the nucleus, stimulates the expression of Hes and He genes, and that changes the behavior of this cell. So this cell becomes a tip cell. This is the leading front of a new blood vessel. That was the first one to receive the VEGF signal, and these become the stalk cells. They are basically the luminal cells. So here's the tip cell. This is the first cell to receive the VEGF signal. That will signal to all of the adjacent cells to not be a tip cell. So we call this lateral inhibition. And this means that when VEGF is received by a bunch of endothelial cells, one of them takes the lead to become the leading cell, and all of the others follow behind to create the lumen of the new microvessel. Now the effect of this is quite profound, and this shows an illustration of the vascular pattern of the retina of a mouse's eye if you start messing around with Deltalike ligand 4. So this is a wild type eye. We've got every now and again we've got vascular branching, so every now and again an endothelial cell has decided to become a tip cell and create a new blood vessel. If we have got low levels of Deltalike ligand 4, so in this case a mouse that's heterozygous for Deltalike ligand 4, there's only half as much Deltalike ligand 4 present. So therefore, when a cell receives a VEGF signal, there's only half the chance, or a reduced chance, of that cell doing lateral inhibition. So you get far more tip cells, and therefore you get a far higher vascular density. So this is a normal situation. You get a normal branching pattern, normal vascular density, with lower levels of Deltalike ligand 4. Basically, loads of cells are deciding to become tip cells, and that creates a really dense vascular network which is highly inefficient. Look at this in terms of veins and arteries. Imagine this is of the two sides of a main trunk road, a road going in and out of a city. You want big roads going in and big roads coming out, and then smaller roads in between linking everything together. This would be like having a city full of roads, but every single road is a side street. The net effect of this is really poor blood delivery to these tissues. Okay, so I'm now going to move on from cell signalling onto some cellular studies of cancer, of both cancer stem cells and normal stem cell populations. I told you earlier on in the lecture that stem cells very rarely divide, and that means that they could then be studied in the lab, working on the basis that stem cells very rarely divide. And this is an experiment that we've done in our lab to study stem cells within cancer populations. So within cancer populations, we think that there is a stem cell-like population in some cancers. They behave like stem cells, and they might be the root of the cancer. So I'm going to take you through this little experiment, and I think Dr. Hayward-Small might have taught about PKH staining of stem cells in a previous lecture, but I'll take you through this. PKH26 is a dye that stains the lipids of the cell surface of cells red. And the only way that cells can get rid of that dye is by cell division, so a cell divides and becomes 50% as red as it was before it divided. So it basically splits the red dye between the two daughter cells. And we can monitor this by flow cytometry, and this is a very bright population of PKH red cells. Grow your cells in cell culture for 5 days, and you see some of the cells have lost their red stain because they have divided lots of times, whereas other cells have not yet really got going. If we allow this to continue for many more days, by day 14 you end up with a relatively rare subpopulation of cells which have not yet divided over the past 14 days, or divided very infrequently. And these red cells might represent a non-proliferating quiescent population. So quiescent is a word that we give to stem cell populations which could be stem cell-like but are choosing deliberately not to divide. They could also be senescent, which are old, tired, damaged cells, which equally will not divide. But by separating out these cells, we can study them and ask questions about whether they are stem cell-like or not. Now, in the experiments that we've been performing, we've been working on multiple myeloma cells. I mentioned multiple myeloma cells in the proteasome lecture. They're very sensitive to proteasome inhibitors, but that's a different story. So what we've done is we've taken some pKh high cells and we've performed FACS, so flow-aided cell sorting. So it's a flow cytometry based technique, and we're sorting out all of the pKh high cells into one well and the pKh low cells into another well and studying them in isolation. And what we found is that all of the pKh low cells are drug sensitive and all of the pKh high cells are drug resistant. And this is a really interesting finding, and it suggests that the quiescent population within multiple myeloma, and this is true for other cancers, is potentially what mediates drug resistance. And we think that these are a stem cell-like population. So this is just one experiment where we are using the feature of quiescence to identify a non-proliferating population and then finding out if they are drug resistant or not. So this is what we call quiescence. We can also study other sort of stem cell-like markers, and again this is in the cancer context, looking at cancer stem cells. And there's a marker called aldehyde dehydrogenase, which is present in stem cells, particularly hemopratic stem cells, but also other tissue stem cells. And what we found is this enzyme is present in a very rare subpopulation of prostate cancer cells that we routinely grow in the lab. And what we did in this particular study down here is used aldehyde dehydrogenase activity to select out what we think are the cancer stem cells. Now, it's very difficult to see in the middle of this picture. This is a big, aggressive, growing colony of cells. And right in the center, about there are a couple of green pixels, which is very difficult to see. You might be able to pull that out, might not. Doesn't really show. I can assure you there's a green pixel within there. And that is a ALDH positive cell, which we think gave rise to this highly aggressive, rapidly proliferating colony. When we isolate the ALDH positive cells and put them into cell culture, they give rise to highly proliferative cells like these, whereas the ALDH low population gives rise to fewer cells. Equally, if we take the ALDH high population, we get more big aggressive colonies when we plate them at clonal density in a cell culture well compared to the ALDH low population. So we think that the ALDH high population represents the stem cell population. Now, as part of that same study, we also identified that within cancer cells there is what we call a stem cell hierarchy. And this stem cell hierarchy is separated into holoclones, mericlones and paraclones. And paraclones are formed where a single cell lands on the plastic of a cell culture dish and grows and forms a small spindly little colony that doesn't have the capacity to grow any further. So if you took these cells, disaggregated them and put them into a new well, these cells just don't grow anymore. You take a larger colony, we call it a mericlone. If you take these cells, disaggregate them, put them into a new well at clonal density, they form more mericlones, which are not particularly fast growing, and lots of paraclones. However, these colonies, these very aggressive looking colonies, much like this one that we've just seen, these are what we call holoclones. And if you disaggregate this contains primitive cells which can give rise to more holoclones which grow very fast, mericlones and paraclones. And the cells that give rise to holoclones are a minority within typical cell lines. And we showed this for prostate cancer cells. So this shows that within established cell lines that we grow in the lab, we have a stem cell hierarchy. Some cells are more primitive than others, and those more primitive cells have a higher proliferative capacity. Now, this has direct relevance to this slide that I showed in part 1 of the lecture, where we think that stem cells give rise to transient amplifying cells which are highly proliferative, and then they go on and differentiate and stop growing. And those differentiated type cells, I think, are what we have got in our paraclones, whereas in our holoclones we have a transient amplifying population which has huge proliferative capacity to give rise to very quick growing transit amplifying cells. And importantly, these cells seem to have the ability to self-renew and maintain their numbers, but they seem to be a quite rare cell within cultured cell lines. Okay, so summary of everything that we've covered. We've covered the Wnt signaling pathway and the Notch signaling pathway. We've looked at how stem cells can differentiate and how these signaling pathways can control cells to develop into one cell type or another. So with the Wnt pathway, we looked at gastric and colon cells differentiating and under the control of the Wnt signaling pathway. Notch signaling pathway, we've looked at vascular differentiation and vascular patterning. When we have a stem cell that differentiates into initially a transit amplifying, rapidly proliferating cell, so they grow very, very fast, and then they start to differentiate and slow down, and then those cells start to do a job. And something parallel to this may happen in cancer, where we have a stem cell-like population giving rise to a transit amplifying population, or maybe just a transit amplifying population but no stem cells. We're not quite sure yet. But these divide very, very rapidly, but then the bulk of the population is actually non-proliferative after a relatively short period of time. We've also looked at some experiments to study stem cell-like features. So the PKH staining is important for identifying quiescence, and quiescence is a feature of stem cells. So quiescence is really a term that we give for cells that are undergoing a deliberate extended G0 non-proliferative phase, but they can opt out of that phase when they feel the need to. So these cells are extended G0 non-proliferating, but then at some point they might come out of that, give rise to a transit amplifying cell, which will then divide very quickly for a period of time, and then sometime later this might do another division like that. So these are very slow cycling cells. These colourless cells here are very fast growing cells. And everything that we think we know about stem cells, a similar process might be going on within cancers. Cancer seems to be a disease of defective differentiation. So we think that cancers contain stem cell-like populations, and basically cancer is a disease where normal tissue differentiation has gone wrong in some way. So whenever we're thinking about, as a cancer biologist, I'm always thinking about what is the likelihood of stem cells resulting in the cancer phenotype that I'm observing. Okay, that's probably more than enough for this lecture. So I will stop the video now.